I'm honored to be part of IMCAS Brazil. Thank you very much for inviting me, and I'm very sorry that I was unable to be there with you in person. We're going to be talking about the exosomes miracle and why it's a double-edged sword. I have no conflicts of interest, and this review is based on about 430 articles. This is the fathers of exosomes. The Nobel Prize in Physiology or Medicine 2013 actually started from 1999 when the scientists found out that another Nobel Prize that there is the cells have these packages that are addressed the way FedEx or DHL address their packages, and they're delivered to their specific addresses. In the 2013, they found the machinery that regulates the vesicle traffic, and that's a major transport system in our cells of how our cells basically communicate. The exosomes are extremely tiny. They're membrane bound and they are 30 to 100 nanometers in diameter. They originate and released into the environment through exocytosis, and they carry proteins, lipids, nucleic acids, microRNAs, messenger RNAs, etc. And all these are involved in gene expression. And you can see here how the early endosome will turn into the MBB, the multivesicular body, and from there it's going to turn into the exosomes. Basically, they're used for signaling. Cell signaling depends largely on exosomes, and it has to do with immune responses, genetic material transfer. They're very important for signaling communication. You can see now exosomes here under the microscope from the liposome. The exosomes are here at the edge, and how this will be eventually transferred into another cell. Exosomes biogenesis depends on fusion with the MVBs. The complex process exosome signaling has cytoskeletal proteins, metabolites, nucleic acids. So you can see here all these different aspects of where these different elements inside the exosomes are carried. Again, exosome is like a package, it's like a FedEx box that carries information in this particular case. And exosomes biogenesis is mainly divided into two ways. There is dependent on the transport of necessary endosome sorting complex, or it's independent of it. And exosomes facilitate cell-to-cell communication. This is what they're for. And how does this happen? Basically, it happens by a fusion of emits with a cell surface released intraluminous vesicles as exosomes, and exosomes may fuse with plasma membranes, or they are taken up whole via endocytosis, or they're delivered to the cytosol via back fusion event, etc. The exosome is a cocktail of non-long coding RNAs. These are involved in gene expression. MicroRNAs are also involved in gene expression, and messenger RNAs that basically carries the information from the DNA to the cytoplasm to compose the different proteins. Exosomes have a role in carrying and spread of proteins, lipids, microRNAs, mRNAs, and you can see again how enlarged here what an exosome can kind of include and different proteins. Cell communication by extracellular vesicle RNA, some known and some have unknown aspects. Now, this is the problem with exosomes, that some of the aspects of exosomes, what they're including and the meaning in cellular communication and cellular interaction is unknown at this point. This is one of the problems. So when we're injecting exosomes, we sometimes inject things that with unknown components that we don't know what are doing there and how the system will respond to them eventually. So they have been very, very important in early detection of cancer and response to therapy and prognosis, and a lot of other aspects of medicine. They have been extremely useful. However, there are preparation problems. As you can see, certain methods have 50% purity, and of course, some of them will be damaged. However, other methods that it's 99% purity, then basically a lot of the function and ability of the exosomes might be lost. They're extremely sensitive. So purification processes are extremely important, and whatever we are told, we're not in the lab to observe them and be certain that what we're told is absolutely real. So there is a design and production of drug-loaded exosomes with a product that target tumors to relieve us from tumors and help us in cancer treatments. And purification and quality assurance of drug-loaded exosomes is very, very important because they can have toxic elements in there, and that will basically affect the system and administration of drug-loaded exosomes. Subsequent inhibition of growth tumor is also very important, that basically they can have the opposite effects. But exosomes have also been used as vaccines very, very successfully, and this is how different processes in different years and different clinical trials and how they have been basically applied. Exosomes have been glorified by anti-aging medicine, promising scar removal, skin regeneration, pigmentation regulation, and hair growth. However, remember, some of the elements that the exosomes include may not be known. So we don't know what is their role in them. They can be good, or it can be bad, or it can be ugly. And we need to be careful because exosomes secreted by keratinocytes can actually increase pigmentations. So all those things we need to know before we start using exosomes. So the beneficial, that's the green part, as you can see in this slide, and potentially detrimental, the red part that you see in this size, are kind of half and half at this point. So you can use it for stem cell plasticity, immune response, and the nervous system. However, the virus can also take advantage of exosomes and utilize them to transfer the virus throughout the system. And so does cancer cells. And so is neurodegeneration. So it's not all good, and we have to be aware of these things. So exosomes have contributed in the development of several diseases, and there's a lot of articles now that are coming on one after the other, explaining this to us. Exosomes have been observed to be involved in spreading pathological misfolded proteins. That means when the protein is misfolded and pathological, that means the whole system is going to be disorganized because the proteins will give nonsense signals, and as a result of these nonsense signals, there will be a problem with spreading of disease or actually enhanced aging. And exosomes have been observed to be involved in spreading pathological misfolded proteins that can actually cause cancer. And that's another article that basically is developed to that. So exosomes have been known to help breast cancer. However, they're also known to increase cancer spread. So some of them can be very good in treatments of cancer, and some of them actually are damaging. They increase the cancer. So exosomes have contributed in the development of several diseases. We have to keep that in mind when we sort of inject the exosomes, and they carry a diverse cargo of proteins, nucleic acid, and lipids, and that can have therapeutic effects or can have pathogenetic effects. So we have to be very important because if the immunity is low, they can have pathogenic effects. So the immunity is high, so it depends on the person. Exosomes carry long non-coding microRNAs and RNAs that basically is a double-edged sword in gene expression, because gene expression can be a good thing, but it can be a bad thing. You can express a gene and become gone, or you can express a gene and basically become schizophrenic. So the main determinant of genetic disease is the expression of genes with harmful mutations. So it can be a problem. Gene expression is not all good. So the expression of the same gene can have both positive and negative effects. There's a lot of articles that show that. And there has been a study with mutated telomerase, the POT1 gene, basically regulates telomerase. And people that have mutations in that gene, they have more telomerase, but however, they seem to have more malignant neoplasms and skin cancers. So longer telomeres are not necessarily a good thing. We don't know enough about all these genetic aspects. So gene expression can be beneficial to the young. The same expression, gene expression, can be beneficial to the young, can be deleterious in the old. And you can see here in the liver and the kidney and the hippocampus, and how in the young is a good thing, and in the old is the exact opposite. It's completely reversed. Emerging roles in exosomes in normal pathological conditions have been in infectious diseases, stem cells, heart, brain, tumor, pregnancy. They have a lot of applications, but we have to be careful because of concerns and challenges related to applications of exosomes in regenerative medicine that may basically lead to infectious spread or to decrease safety. So we have to be careful. It's not all good. Exosomes therapy may serve as a new approach to combat aging. The translation of preclinical results to clinic needs more extensive investigation on exosomes, both in their biologically and related techniques. There are new technologies now, first developed in 2020, signaling resonance energy transfer, that could enhance the transport of exosomes to the right address. Basically, can direct them properly. And this is how basically what it is and how you can see it under the microscope, how it basically one cell can affect the other, and how it can target the exosomes to the cell that needs it the most. And you can help by, which is very important, because if the exosomes has misfolded proteins, then you're going to have problems, not only with aging, but with increase of disease. And with this, thank you very much for your kind attention. And if you have any questions or concerns, please get in touch with me at science@iellios.com. Thank you very much. This is my WhatsApp. You can take a picture. I'm there to answer any questions you may have.